present Dr. John Demuth to talk about his research today, and he's a professor and associate dean for research in the University of Louisville School of Dentistry in the Department of Oral Immunology and Infectious Diseases. A little bit of background about Dr. Demuth is that he's an oral microbiologist who studies the mechanisms that contribute to the formation and development of microbial biofilms. And the long-term goal of his research is to develop approaches that can control these processes for the treatment of common oral diseases such as periodontitis. He obtained his Ph.D. in molecular biology from Lehigh University and subsequently completed his postdoctoral research at the Wistar Institute in Philadelphia. He was a faculty member at the University of Pennsylvania School of Dental Medicine from about 1986 to 2003, and currently in the role that I described. And it's my pleasure to introduce him as we are working on a collaboration to create nanoparticles to target oral biofilms. So with that, I'll let you.
Thank you. It's a pleasure to be here. Only one correction. Actually, I'm here to talk about our research, not just mine. So I'm in the School of Dentistry, and as such, one of our main focuses and the systems that we work with is the oral biofilm. All right, so just a couple of introductory tidbits about the biofilm. It's probably one of the best characterized microbial communities that's associated with humans. Right now it's a very complex community. There's probably over 700 different bacterial species that are known to reside in the oral cavity. In addition to that, it harbors fungi, bacteriophages, and since the oral cavity is an open system, open to the environment, there are transient organisms that aren't resident. And that probably accounts for some of these studies that show the complexity of the community is even higher than 700. There's papers that say 1,500, 2,000, 5,000 different species. These are probably transient organisms. That said, if you just sample a given person, say in this room, and ask who's in that biofilm, about 90% of the biomass is accounted for by 200 to 250 species, and the rest are minor components. And it's actually not just one community, there's multiple communities. There's a different community on each tissue surface. So the buccal epithelial will harbor one community. The surface of the tongue, a different community. Supragingival surfaces of the teeth will be different than subgingival. So there's a lot of different subcommunities within this biofilm that we collectively call the oral biofilm. And it's a highly structured, highly organized community, and in this case, pictures are more than 1,000 words. So here's some images of the oral biofilm formed on natural teeth. These are images collected by fluorescent in situ hybridization techniques. Most of these images are hybridized with multiple probes. So for example, over here you have a probe in red that is relatively non-specific. It reacts with 3 different groups of bacteria, the CFB cluster, and then it's counterstained with a probe that is specific for the genus Prevotella. And so what you can see is that these Prevotellas form microcolonies, almost look like little flames on the top of a candle that sits on top of this mass of CFB organisms. This is the organism that we study. It's called Porphyromonas gingivalis, and you can see that it's growing within this mass of CFB bacteria, but forming these stalk-like structures, probably because it's associating with filamentous organisms within this CFB group. But my favorites are here. Look at this structure. It's like a test tube brush. You have the core of the brush, or these organisms here in the center, lactobacilli, and then CFB organisms all around this core, like the bristles of a test tube brush. So this is the community that we're dealing with. Very complex, highly structured and ordered community.
What is the size on that steel bar, Don? Just to get a sense for what we're looking at.
Okay, I'm trying to remember.
We're talking about 100 microns or more, or it's many cells, or many bacteria.
Yeah, this is probably 20 to 30 microns in that image there. Okay, so these are kind of macro structures, but depends on how you define micro and macro.
That's perfect, yeah.
Okay.
Macro, or microstructure.
All right, so, and the reason that this is important is that the common oral diseases, caries and periodontal disease, are biofilm-mediated diseases. And you don't get these diseases by becoming infected with a pathogen. The pathogens are already there and resident. Oral diseases arise when the natural balance or homeostasis between microbe and host is disrupted. And as a result, you have changes in biomass, changes in the populations of bacteria, and it's those changes that induce the inflammation associated with periodontal disease. So we call these like kind of ecologic catastrophes. So oral diseases don't necessarily follow Koch's postulates. And this goes back, I think, probably over 20 years that the association between different populations of bacteria and health and disease was apparent. And so this is a summary of a study that was done up in Boston about 23 years ago. They were simply trying to catalog oral bacteria, and what they found was that they associated with groups. And so if you find Villanella, you usually find Actinomyces nearby in the same niche. Same with the Streptococci, and you can follow through the other colored boxes. But the second observation that came from this study was that when they looked at microbes collected from a healthy individual, the predominant organisms were in these first three boxes. And when they looked at microbes from a diseased individual, these organisms predominated, the orange and the red. And so the red complex are three species that are highly associated with chronic adult periodontitis. So there's, again, the first demonstration of some type of a correlation between populations of bacteria, health, and disease. Okay, so how do dentists deal with biofilms? It's still a rather primitive approach. It's a global approach. They scrape it off. And if there's any damage to the tissue, then they repair or restore what they see. And the tools of the trade that they use, everyone I'm sure is familiar with. You may think they're more like medieval tools of torture, but these are the tools that dentists use to remove biofilm. And in terms of prevention, what do they advise? Go home, brush, and floss. So the tools to prevent oral disease that we have available are limited to those right there. So this is a question that I ask incoming dental students. And I think this is the important question that really forms the basis for a lot of the work that we're doing in the laboratory. Is the presence of the oral biofilm necessarily a bad thing? And when I ask that of the D1 students, they're squirming in their seats because they think the answer is obvious, but why would I be asking the question if the answer is the obvious one? So my answer is no. And the reason it's no is because most of the bugs that one finds in the oral cavity are benign. They're commensal organisms. Some may actually be beneficial, symbionts that help in one way or another. So the question becomes, why remove everything? Why not just target the ones that are most associated with disease? And there's a growing literature that suggests that that approach is feasible to control oral diseases. And probably the seminal paper is this one here, done by a colleague of mine, George Hodgson Gallus. Low abundance biofilm species orchestrates periodontal disease through the commensal microbiota. So George was interested in studying virulence mechanisms of one of those periodontal organisms, Porphyromonas gingivalis, or PG. And so they infected Baupsi mice wild type with PG. And what they saw was that the infection led to a high and chronic inflammation, which resulted in tissue damage and bone loss, which is the clinical presentation of periodontitis in humans. Well, they probably should have just stopped there, but they said, well, we could probably learn even more about virulence mechanisms if we put PG in a mouse that doesn't harbor indigenous bacteria. So they did that. Now here's the pathogen infected into a germ-free animal. The result, no symptoms at all. And so this was a head scratcher for this group for a while. And the reason that they finally identified for that behavior was that what PG is actually doing when it enters the biofilm, it's only a minor component in the wild type, but its presence induced profound changes in both the overall biomass of the indigenous organisms as well as populational changes in that community. And it was those changes that induced inflammation and ultimately led to disease. So they coined this term, P. gingivalis, as a keystone pathogen. It's a keystone organism. Well, the keystone concept isn't foreign. It hasn't been used in dentistry until recently, but it's known in ecology. And so if you go to the ecology literature, they define the keystone organism or species as a plant or animal that plays a unique and crucial role in the way an ecosystem functions. Without it, this ecosystem changes or ceases to exist. Or you can look at it from the other direction. A keystone organism enters a community and changes that community in one way or another. So we don't have to change many words in that definition from the ecology literature to define what P. gingivalis was doing in the mouse. A keystone pathogen is an organism that plays a unique and crucial role in the way a biofilm forms and functions. right? And its presence or absence can induce changes in that community. Okay? So this is really the basis for targeting specific organisms to control a disease that's associated with a very complex community. And there's a growing literature, as I said, that is now suggesting that this is a feasible approach to control disease. So this is what we study. Our target organism is this periodontal pathogen, P. gingivalis, or PG. Its natural and ideal niche is in a subgingival pocket under the gum line. And when we started this work years ago, we started with a really simple and maybe naive question. How does PG get there? Like all oral organisms, PG is transmitted by human to human contact, so it enters from the outside. And so the first environment that PG experiences is not its ideal subgingival niche, it's the supragingival environment, which is not really conducive for PG to grow because it's aerobic and PG is an obligate anaerobe. So how does this organism overcome these challenges? Well, it interacts with a pre-existing supragingival microbial community. And it's these interactions that were of interest to us, because they're some of the first things that occur once this organism, PG, enters the oral cavity. And that's of interest because they're ideal points to intervene, because if you can stop those first interactions from occurring, all the subsequent downstream steps that need to occur to become established in this ideal niche where it causes disease, they become moot. So this is what we were studying. We began to study some of the first things, first interactions that PG undergoes once it enters the oral cavity. And to summarize maybe 5 years of work in a single slide, what did we find? First, we know that P. gingivalis adheres to oral streptococci, some of the primary colonizers of the supragingival biofilm. And that interaction is a protein-protein interaction that occurs between the minor fimbrial antigen protein, this is the MFA, of PG with a protein on the streptococcal surface called SSPB. It's a member of a large family of proteins collectively referred to as antigen 1-2. And we also determined that this interaction of P. gingivalis with antigen 1-2 is selective. In other words, PG doesn't interact with all oral streptococci, only certain species. And that's interesting because this antigen 1-2 protein is highly conserved across all streps. So PG is selectively identifying certain members of this antigen 1-2 family and interacting with the organisms that express those members. And that's really illustrated here. This is just an in vitro binding assay looking at PG adherence to two different purified antigen 1-2 proteins, SSPB, which is a functional protein, it binds to PG, and you can see that here, and SPA-P, which is an antigen 1-2 from strep mutans, which does not interact with the PG, and it doesn't bind very well at all to that antigen 1-2. Now, if you compare those two proteins, they're very similar. This is the active protein, this is the inactive protein. Primary sequence identity, over 70%. Conservation of structural organization. Both of these proteins have these alanine and proline repeats, have a C-terminal region, cell wall anchoring. The only thing that really differs in the proteins is this central region, the divergent region. But if you were to assume that the functional differences are derived from that divergent region, you'd be wrong. So we were interested in how PG selectively identifies a protein like SSPB and ignores this highly related antigen 1-2 like SPA-P. And to answer that, we actually took advantage of the structural similarity within this family, and we constructed a series of chimeric or hybrid proteins that had increasing amounts of N-terminal sequence derived from SSPB fused to the corresponding C-terminal regions derived from this inactive S-mutans protein. And then simply asked, how many of these hybrid proteins are functional and bind to P. gingivalis? And the answer is only one of them, SP5. None of these other chimeric proteins were active. And the difference between SP5 and SP4, active and inactive, is only a very small region of the parent protein. And we designated that region as BAR, for SSPB adherence region. And the graduate students get a kick out of that because then any experiment with a negative control they call bar none. And if we have to increase the concentration of bar in an experiment, they say we're raising the bar. Okay, so we think at this point we've got it. Because all we need to do to understand this functional difference is to compare this region of SSPB with the corresponding region of an inactive protein, and it will become obvious. Wrong again. So here's a direct comparison of primary sequence between these two regions of active on the top and inactive protein on the bottom. And I don't know, maybe you're more astute than I, but when I looked at this, there was nothing that really stuck out and said, well, this is why this protein is functional when another one is not. So we went one step further, and we predicted secondary structure. And even here, we kind of scratched our head and they said, well, this looks pretty similar up and down. There was just really one thing that we noted, and that was in the N-terminal region, that in the functional protein, this predicted helix transits into a beta sheet, and then the non-functional protein, this predicted helix is interrupted with a beta term followed by a second alpha helix. The reason that these two sequences predict differently is the presence of this glycine in place of asparagine and proline in place of valine, and that gives us 3 helix-breaking residues in this cluster of 6, and the programs predict that to be a term. So is that functionally relevant? Well, to answer that, we made synthetic peptides representing each of these 2 regions, and this is their behavior. The peptide 27-mer derived from SSPB interacts with PG very well. The corresponding region from SPA-P does not. In fact, this 27-mer here interacts with PG as effectively as the entire parent protein of 1,500 plus residues. So all the binding activity of the SSPB polypeptide is accounted for in this 27 amino acid structure. And the reason for that becomes apparent when we looked at this sequence in more detail, because it actually represents the first prokaryotic equivalent of a known eukaryotic protein-protein interacting domain called the nucleoreceptor or NR box. All right, so here's a comparison, a bar and an NR box. The nucleoreceptor box has a core element off the helix with a consensus of leucine, XX, leucine, leucine. That's present in the bar sequence. That core element in the NR box is flanked by polar or charged residues that help stabilize the interaction of the nucleoreceptor with the coactivating protein. Those polar residues flank this VXXLL sequence in bar. And then there's a sequence in the NR box that can reside either upstream or downstream from this core element, which drives specificity. It dictates what coactivating protein this nucleoreceptor binds to. And we think that that specificity determinant is NITBK in bar. And I don't want to show you the data, but we've gone on to prove that by doing some site-specific mutagenesis, introducing glycine for this position and proline for this position. These are the residues that the inactive protein has, and each of those single substitutions renders the intact SSPB protein inactive. Okay, so one change changes the functional activity of a big polypeptide of 1,500 residues and inactivates it. And to further confirm, now that we have multiple genome sequences, we can go in and ask how common is this combination of VXXLL and NITBK amongst strep's. And it turns out it's present only in these 3 organisms, Ascordonii, Oralis, Sanglums. Each of these organisms interacts with PG. You don't have this combination in any of the other strep's in the current database. PG doesn't interact with any of these other organisms. Okay? So what we know is that interaction of PG with Orostrep requires a motif that resembles this nucleoreceptor box, and the specificity of interaction is driven by this NITBK motif. At this point now we can start asking more translational questions. Can we use these peptides as potential therapeutics to block the colonization of oral cavity by PG? And to look at these types of processes, we have a couple of different experimental approaches that we rely on. A in vitro biofilm culture model, actually a simple one of 2 species, just involving S. gordonii and PG. And then we can make it more complex by adding additional oral organisms. And then we have a mouse model of periodontitis, where we can look at the function of these synthetic peptides in vivo. So here's a result of one of those experiments. This is our 2-species biofilm. We visualize it by confocal microscopy. So we label the streptococci with the red fluor, hexidium iodide. We allow them to adhere to a cover glass, wash away unbound cells, and then add PG, which is labeled with an Alexa 488 or similar fluor. And so PG adheres to the immobilized streptococci and starts to accumulate in these microcolonies, shown in the green. If you do this in the presence of bar peptide, this is what you see. So a dose-dependent reduction in green fluorescence arising because PG is unable to adhere to the streptococcal substrate in the presence of peptide. And actually, this is a fairly potent inhibition. IC50 of about 1.3 micromolar. We see similar results in an in vivo model. So here's the general protocol that we use for an animal model. The mice start off with an antibiotic treatment to knock down the indigenous community, and a washout period, and then we infect first establishing strep gordonii in the oral cavity, and then following up and challenging these animals with P. gingivalis in the presence or absence of peptide. And this is what we found. So all the data here are normalized to sham-infected animals. So by definition, sham-infected animals have no bone loss, and I should mention that's our outcome here. We're looking here at day 70 at alveolar bone resorption, which is the clinical presentation of periodontal disease in humans. So we're measuring the loss of alveolar bone. If we infect with only strep, you see that these animals are not much different than sham. If you infect singly with PG at a low dose, it might be trending towards seeing some bone loss, but this doesn't reach statistical significance. But it does if you infect with a high dose, 10 to the 9. If you have strep present and then challenge with PG, even the low dose of PG shows significant bone loss. So the presence of streptococci facilitates PG colonization and virulence. And then if you do that in the presence of bar peptide, this is what you find. These animals are no different than sham. So the function of this peptide occurs, you can see it both in vivo and in vitro. And it appears to be a potent inhibitor and a potential compound that could be used and developed as a therapy. So how can we apply, where are we going with this, essentially? How do we apply this approach to human disease? So what we envision is something that is topically applied, like most oral healthcare products are. Maybe formulate this peptide in a mouth rinse, a toothpaste, or a dental varnish. And the purpose would be, ideally, to limit the recolonization of the biofilm after professional cleaning. Because one of the limitations of the current gold standard approach to treat oral disease is if the dentist removes biofilm, it starts to reform almost immediately. And so our intent is to let it reform, but under an environment that prevents PG from joining in. And so hopefully we can direct the new biofilm that forms into one that's in balance with the host rather than a dysfunctional organ or community. It can also potentially be used to reduce existing biofilms. So rather than target the early stages, maybe it is also effective at eliminating mature communities. So if we think along those lines, there's probably a couple of limitations that you can come up with. Most of you brush, swish, day and night, right? So you're looking at transient exposure. At best, maybe 30 seconds, 2 times a day. Well, that raises some significant questions. Will that deliver a sufficient dose of peptide to be effective over that 12-hour period between brushes or swishes? Is that transient exposure long enough to be effective? And we also know that disruption of mature biofilms requires much higher concentrations of peptide. So it's harder to eliminate an existing biofilm than it is to prevent one from forming. So how do we address these issues? And this is where I went knocking on Jill's door and said, I need some help here. I need to find a way that we can deliver higher local doses of peptide to a given site, and I need to find a way that maybe we can overcome this transient exposure and deliver our active agent over a broader window of time. And so she says, I can help you out with that. We have these nanoparticle drug delivery vehicles. And so we started with two approaches here, to develop drug delivery systems that would address some of these limitations or potential limitations that we were anticipating. Approach one was to develop modified PLGA nanoparticles. And the goal here is to deliver higher local doses of peptide and hopefully increase effectiveness against established biofilms. The process is relatively simple now that I understand it. When I first started this, I said, Jill, I'm a microbiologist, not an engineer. Tell me what's going on here. She said, well, you take these agents and you mix them up, and it's like making a vinaigrette salad dressing. You shake it real hard, and it emulsifies, and there you go. So now I understand. And if we do that in the presence of an acylated avidin, we can decorate the surface of these particles with avidin, and it's easy then to synthesize a derivative of bar that's biatenylated at its end terminus, and we can stick bar on the surface, and voila, we have multiple bar molecules on a given nanoparticle. So if we do that, we did some physical characterization of the resulting surface modified particles. We looked at unhydrated and hydrated diameters and looked at the zeta potential, or the surface charge. What you see here is modification. There's a trend here towards smaller particles, but that's a trend because that didn't reach statistical significance. When you hydrate them, they're all about the same. They increase in size, as you would expect, all to about 300 nanometers in diameter. The particles unmodified, because of the nature of the polymers that are used to make them, which are negatively charged, has a negative zeta potential. And as we modify and add positively charged avidin and bar, that negative charge is reduced. Okay, so this is just a way to follow that we're modifying the particles appropriately.
You unhydrate them, you just dry them out? Because you make them in liquid state, or is it a
Yeah,
salt concentration issue?
Essentially lyophilized.
Oh, okay.
Okay.
For stability, you should keep them in lyophilized form instead of just bring up all the nutrients.
Okay.
Follow up on
Yeah.
that. So when you're saying it's hydrated, is it the actual PGLA that's swelling, or is it going to be the suspension of the avidin and bar as far as making the diameter larger?
So you see that same increase in diameter just for blank polymer alone. So it's the polymer swelling apart is what you want.
And thank you for asking the appropriate individual.
What he says.
Okay, so next we wanted to know how much peptide are we actually getting on these particles? What is the payload? And for that we used a fluorescent tagged peptide and just incubated particles with increasing concentrations of input bar and followed fluorescence as it was associated onto the particles. And what you see is this saturation curve. The particles are saturated really at an input bar concentration of about 37 micromolar, and the saturation point is about 7.5 nanomoles of bar per milligram of nanoparticle. So that's about as high as, under these conditions, synthetic conditions, that's as high as we've gotten in terms of payload. Is it functional? Well, next we asked whether these nanoparticles that are decorated with bar were able to interact with the target organism, PG. Again, a simple in vitro binding assay, and you can see that as the amount of bar and P's increase, fluorescence associated with the surface of P. gingivalis increases. So immobilized on the surface of these NPs, bar peptide is still able to interact with its target. And then finally, is it functional? Can these NPs inhibit P. gingivalis colonization, inhibit that biofilm formation that we see in our culture model? And so I'll remind you again, this is bar peptide alone, soluble, and you see that it's dose-dependent inhibition of PG adherence with an IC50 close to 1 to 1.3 micromolar. When we use equimolar or equivalents, molar equivalents of peptide delivered by the nanoparticle, this is what we see. So at almost every point, you see less fluorescence in the samples treated with bar NPs than we do with soluble bar peptide. So these particles are actually more potent by a factor of almost 7-fold. So if we quantitate fluorescence and plot that out, here's the plots. These are bar NPs. That IC50 here is about 0.2 micromolar. This is bar with an IC50 right at about what we said before, 1.3. So almost a 7-fold increase in potency. And we ascribe this, at least now, to increased valency of interaction of particles delivered by, or a bar delivered by particles to the target. And we actually have a student that's actually trying to demonstrate that experiment. Okay, so surface modified NPs, what we have right now are about 7-fold more potent than free peptide. It interacts probably with PG intervals at higher valency, so the avidity's increased, and we're trying to establish that experimentally. We have another student that's looking at established biofilms. Are these bar NPs more effective at disrupting an established biofilm than soluble peptide? I don't really have any data right now to do that. But we still haven't addressed this transient exposure issue. These particles, if we were to formulate into a rinse or a toothpaste, are still only going to be present for 30 seconds before you rinse them out or spit them out. So the second approach that we used was to try and develop nanoparticles that would address this transient exposure issue. And that approach involves generating targeted sustained release nanoparticles. Okay. So the goal here is to target these nanoparticles to the niche that PG seeks to occupy in the oral cavity. And that these nanoparticles are encapsulating bar peptide and releasing it for a broad or a longer window of time. And our target characteristics would be to have nanoparticles that are retained and release at least an IC50 concentration of peptide over a 12 to 24-hour window. That way, swishing in the morning, if you can retain that nanoparticle in the oral cavity and it's releasing peptide over 12 hours, you have constant exposure to the active agent until you swish again in the evening. Okay? So this just schematically is illustrating the strategy here. We said that PG enters the oral cavity and interacts with the supragingival biofilm, like that. What we want to do is target a nanoparticle to that same niche. So when PG comes onto the scene, first its niche is obstructed by a nanoparticle that's secreting or releasing an active agent that inhibits PG adherence to this community. All right, so it's done basically the same way, except we have bar present earlier on in the synthetic reaction, so that these particles encapsulate bar as they form. We've done that, and I'll show you some data on bar loading and release. The second step, which we haven't completed yet, is to add this targeting molecule. Surface modify these sustained release particles so that they're targeted to the correct niche in the oral cavity. And our targeting molecule is a cafe protein. This is a protein that is expressed by another oral organism, Actinomyces auris. Remember, we said that oral organisms tend to cluster. You find them in groups. And A. auris and Strep gordonii are compatible. You find them in the same niche. In fact, auris adheres tightly to Strep gordonii through the action of this cafe protein binding to a surface carbohydrate on the streptococcus surface. So it's an ideal targeting molecule. And we have actually two approaches that we're going to look at to surface modify these sustained release particles. One is to acylate cafe directly, so it is incorporated onto the surface, much like Pathogen was in our previous example. Or the alternate approach is to follow that intermediate step like we did in our first, where we interact, or we mix it with acylated adenine first, and then maybe biotinolate cafe and have it attached to the surface adenine that's present. All right, just some characteristics of these sustained release. First question, how much payload are we getting in there? So if we form these particles in the presence of 43 micrograms of peptide per milligram of polymer, over at least the first 2 experiments that we've done, we're consistently seeing about 33% incorporation. So total payloads are roughly 14 micrograms of bar per milligram of NP. And we've done some preliminary work on release kinetics from these particles. And that's expressed here as both peptide concentration released or percent peptide that's released. What we find is that our current particles, about 75% of the encapsulated peptide is released during the first 4 hours. So we're not quite there yet in terms of the characteristics that we desire. We'd like to see release out here to 20 to 24 hours, or 12 to 24 hours. So in essence, these particles would be effective only for the first 2 to 3 hours. After that, they're releasing less than an IC50 of bar into the environment. So we need to optimize this, and that can be done in a number of ways to try and, one, increase the payload and lengthen the release profile. Other things that we're going to be working on in the near future is to optimize CAFE surface modification, and look at retention kinetics. How long do these particles stay on the surface of strep? And then ultimately test in vivo and in vitro for inhibition of P2 adherence. And then one last thing that's ongoing now, in some early studies, we're looking at not particles, but nanofibers, and asking, can they be developed as sustained release vehicles? And what we know so far, at least with one student who's worked on it, is that particles can be loaded, or fibers can be loaded with bar peptide, but they're releasing very low levels. So we still have some issues to deal with on that. And I still have 5 minutes or so maybe I do have time for shifting gears one more time. There are additional limitations that we have to consider. Using peptides as therapeutics. Peptides are expensive to synthesize. Well, that cost to benefit ratio might be acceptable if the peptide was targeting a disease like cancer. But it's probably not acceptable targeting periodontal disease. I mean, no one's going to want to go and buy a $65 bottle of mouthwash when you can get Listerine for $4.95, $3.95 with coupon. So that's a problem. Second, obviously peptides are susceptible to proteolytic degradation. That's an issue with oral diseases because a lot of the periodontal organisms are highly proteolytic bacteria. So there's a stability concern and a cost concern in the synthesis of this active agent. And so how are we addressing these two issues? With another collaboration, this time with a medicinal chemist in the Department of Chemistry. And what we're doing is designing and synthesizing small molecule mimetics of VAR. And the approach that we're using is a click chemistry strategy, where we first synthesize small molecule mimics of each of the 2 functional motifs of peptide, and then we join those 2 small molecules together via the click reaction. And the basis for doing this comes from our knowledge of the structure of this VAR region in the SSPB protein. So we can, on one hand, do some molecular model. A quick example here, this is a model of the VXXLL helix, looking down the longitudinal axis of that helix. And the chemist will look at this and say, look at these side chains of the leucine residues. They're at the corners of an equilateral triangle. And the chemist will then go back and try and generate a small molecule that will position hydrophobic groups in that geometry. So that might be one of the characteristics of one of the compounds that he seeks to generate. There's also crystal structures of the SSPB protein and of other NR nuclear receptors. So we know 3-dimensional structures, and if you're one that's adept at crossing your eyes a little bit, this is a stereo pair, and you can look at it in 3 dimensions if you like. This helix is our VXXLL, NITVK is this red strand that comes down like so. So we have structural information that the chemist can use to start to design small molecules with the physical and structural properties that we see here. And I'll tell you, it was hard enough understanding the engineering. Now this guy is dragging me back to my undergraduate Morrison and Boyd days, organic chemistry. And I had to relearn what's a triazine, what's a pyrimidine. So this is just some examples. Here's one of the end products, a mimic of VXXLL. And remember that model that had the leucine R groups at the corner of an equilateral triangle. Well, here they are. And you can make that triangle between carbon 1, 3, and 5 of this pyrimidine ring. Okay, so the chemist is trying to mimic that structure on a small scaffold, in this case a pyrimidine or a triazine. Both of these are planar rings, and so that the hydrophobic groups are going to be off in the same plane as the ring. We can do it on a non-planar ring like a cyclohexane, and that can put R groups above, below, or even a combination, that above or below the plane of that ring. And then all of these VXXLL mimics have this reactive group here, a carbon-carbon triple bond, or an alkyne. And the reason for that will become apparent here in a second. For NITDK mimetics, we have an oxazole. We've been using an oxazole scaffold, which has certain advantages, like chiral centers, which again can be used to position our functional groups above or below the plane of the scaffold. And these mimics have a reactive group, this azide moiety right here. Once we've synthesized small molecule mimics of each of these motifs, we can bring them together with the click chemistry reaction. And click chemistry reaction is a reaction between an alkyne and an azide. And under the right conditions, these two groups undergo a cycloaddition to generate this triazole ring, which covalently links mimic 1 to mimic 2. And so this is our final product here. So we just had a paper accepted that describes about 80 of these compounds. I'm not going to show you all of them, but this is one of the tables that came from that paper that looks at probably 20 or so. And what we found was that these compounds fell into 3 groups. Some were fairly potent inhibitors of P-gingivalis adherence, and we defined that as an IC50 of less than 10 micromolar. There was a second group that were moderate inhibitors. It showed IC50s between 10 and 20. And then there were poor inhibitors that showed IC50s over 20. So we have in this group right here probably 2 or 3 of the most or more potent inhibitors. And with these compounds, we went and did some additional work to demonstrate that the compounds weren't acting as an antibiotic. We wanted to prove that they're inhibiting this interaction between this interspecies interaction rather than killing the organisms. So we tested essentially their growth inhibition on planktonic cultures of each of the organisms that we use in our biofilm culture. And for each of these most active compounds, we saw no growth inhibition at all. So they're not functioning as antibiotics, they're not killing, preventing the growth of the bacteria, but they're interfering with the interspecies interaction that needs to occur to form that community that we visualize. So, right on time. I can quickly summarize then. We know from the biologic end of things that the bar region of SSPB is a potent inhibitor of P. gingivalis adherens destruct, and it reduces P. gingivalis virulence, and that suggests to us that bar could be a useful therapeutic to limit colonization of the oral cavity and potentially control periodontal disease. We've got proof of principle for the development and use of surface-modified and sustained-release nanoparticle drug delivery vehicles that allow the delivery of higher local doses of peptide for more prolonged periods of time, and these vehicles may actually be the ones that facilitate our formulation into a rinse, because now if we can take those sustained-release targeted particles, you swish with them, the particle adheres to the existing biofilm on the tooth surface, is retained there for hopefully 12 hours or more, releasing active agent. So that by the time you swish in the evening, you've had continuous exposure to the active agent. And then lastly, active peptidomimetic small molecules have been designed and synthesized. They show inhibition in the micromolar range. And in essence, these may be more stable because they're not susceptible to proteolytic degradation, and certainly more cost-effective to produce in large quantities than a peptide. So with that, let me just thank a bunch of people that have been involved with the work. These are people at one time who were in my laboratory, former graduate students. Julie Tan's currently a person that's doing the functional analysis of the small molecules, a long-term collaborator with Vermont. We want to thank Steinbach Rankin's lab and 3 master students who have done a lot of the nanoparticle work, Pariti Kalia, Ranjith Radhakrishnan, and Ankita Jain, and my collaborator in chemistry, Fred Lutzio, and his postdoc, Parvin Patel, who's done all of the organic synthesis. So thank you for your attention, and it was a pleasure to be here and tell you about what we're doing here today.